Okay, welcome back. We are now going to talk about active transport. So, active transport is kind of similar to facilitated diffusion because it is protein mediated. But the big difference here is that we are moving up the gradient. So, this might be a cell that, like, here is these little bit of nutrients outside the cell, and it's pulling them inside the cell where it already has a lot more. It's going against that gradient. So instead of spreading out like people in an elevator, it's packing them into one side. Because we're doing that, it is going to take energy to do that. So in active transport, we're moving things up their concentration gradient, packing them closer together. And because it takes energy, you have to be like forcefully pushing things through them. These transport proteins are sometimes called pumps. This active transport is going to have some sort of exergonic process and the endergonic process coupled together. So to be able to force something to move against its concentration gradient, you're going to have to use energy. A lot of times that energy comes from ATP hydrolysis or breaking that ATP down, releasing the energy, which forces that molecule to move. There's three big reasons why active transport is important. One is to bring in nutrients. No matter if nutrients are scarce in the environment, you still want to bring them into your cell. One is to remove waste. Your cell doesn't really care if there are a lot of waste products outside. It's got to still get those waste products out of your cell. Things like carbon dioxide or such things. And then maintaining ionic concentrations on either side of the membrane. So we want to have a positive charge and a negative charge on either side of that membrane. Even though it's not at equilibrium, we're going to spend energy to do that. We're going to talk about some of these different situations, some specific transport proteins that are involved in all of these. So active transport lets you create an environment inside of your cell that is stable, that's not going to be changing whenever the environment changes. Your cell can maintain its homeostasis and be different from its environment so that the cell has its optimal environment inside. So one big difference between active transport and diffusion is the direction of transport. In diffusion, whether it's simple diffusion or facilitated diffusion, it's really non-directional. It can bring things in or out. It all depends on concentrations. The concentration of materials is the only thing that determines that. But active transport has an intrinsic directionality. We are always bringing in the nutrients. We are always sending out the waste. We are always keeping that movement one way. Whether or not concentrations change, that transporter always moves things one way. We can divide our active transport mechanisms by what source of energy it has. So whether we're getting energy from ATP or we're getting energy from another kind of molecule, and whether there are two solids being moved at the same time. So active transport can be both direct and indirect. In direct active transport or primary active transport, you're forcing things to accumulate on one side of the membrane, like these hydrogen ions here, and that is coupled with ATP hydrolysis. So this hydrogen, even though you only have a little bit of hydrogen inside the cell, we are going to pump it outside. And by pumping that hydrogen outside, it is going to take ATP. So this would be an transport ATPase, transport because it moves things, it transports them, and ATPase because it breaks down ATP. That ACE tells you you're breaking down. So this is a transport ATPase, or sometimes they're called ATPase pumps, breaking down ATP to pump. In indirect active transport, we're taking two things transporting at the same time. So this would be an example of indirect active transport. In this indirect active transport, one thing is moving down its gradient. So here hydrogen is moving down its gradient from where it has a lot to a little. And that allows another molecule to move up its gradient. So this S molecule is being packed together up its concentration gradient. But we're able to pack that S molecule, bring it into the membrane, not with any ATP. We're not burning any ATP on this enzyme, but by bringing this hydrogen ion, by bringing something else down its concentration gradient. So here this indirect active transport is a symporter. It's bringing two things in. But you can also have this type of transport with an antiporter, moving something out of the cell against its concentration gradient, bringing something into the cell down its gradient, or the reverse of that. So two things coming in at the same time. That's an indirect active transport. There's 4 types of transport ATPases, this direct type active transport we have. There are P-type, V-type, F-type, and ABC-type. And those 4 types of transport ATPases, they're all doing active transport. They're all using ATP to give it the energy to it. But they're different in terms of their structure. They're different in terms of how they work, where they're found, and what they do. P-type ATPases are a big family, and they're reversibly phosphorylated by ATP on aspartic acid. And there are 5 types of P-type ATPases. P1 ATPases are found in all organisms. They transport heavy metals. P2 ATPases maintain gradients of ions across the plasma membranes of your cells. So they keep sodium, potassium, hydrogen ions, calcium ions in their proper gradients across your cell membrane. In this role, they're important in muscle contraction, allowing sodium and potassium to move through so those muscles can contract. Also allowing that hydrogen to come into the lumen of your stomach to acidify your gastric juices. P3 ATPases are found in plants and fungi, and they pump protons out of their plasma membrane to make their environment more acidic. P4 ATPases pump cholesterol and fatty acids. They're a type of flipases. They flip those across the bilayer. And P5 ATPases, we don't really know much. You get to be the scientist that figures this one out. Some of them do transport cations. So here's what a P-type pump is going to look like. Next is our V-type ATPases. V-type ATPases are important for pumping protons into organelles. So you find these in vacuoles, in vesicles, lysosomes, endosomes, and the Golgi. And they have these 2 kind of subunits to them. This integral component that's embedded in the membrane, and this peripheral component that juts out from the membrane. F-type ATPases are found in bacteria, mitochondria, and chloroplasts. They also have 2 multi-subunit components, a transmembrane area and a peripheral component. And the peripheral membrane area is where you bind the ATP. And these also are able to work in reverse. So you may have heard of them as ATP synthases when you are talking about like the electron transport chain and how your mitochondria make ATP for you. So if it allows hydrogen to flow down its concentration gradient, it's going to spin around and make ATP. But if it is pumping hydrogen against its concentration gradient, it's going to hydrolyze ATP back to ADP. So this is a really important thing that ATPases can do because they can function both ways. Sometimes ATP can be used as an energy source to make an ion gradient. But also, sometimes ion gradients can be used as an energy source to make ATP. That ion gradient that's used to make ATP is how our mitochondria in the electron transport chain creates most of the ATP that we get from the food we make, from the food we eat. And this concept that ATP can use to create an ion gradient, but an ion gradient can be an energy source to make ATP. This is true of all eukaryotic cells and even most prokaryotic cells. Then we have our ABC-type ATPases. ABC stands for ATP binding cassette. So ABC-type ATPases are sometimes called ABC transporters. And that cassette just is a weird word to describe this region that binds to ATP. ABC-type ATPases are a large family of transport proteins. You find them in all kinds of organisms. Originally, the first ones we found were importers, so they imported molecules or brought them in. But now we know a lot of them that are exporters, and a lot of them are very medically important because they can pump drugs out of cells. So if you have a tumor that has this ABC transporter, it makes it resistant to drugs. This is called the MDR transport protein, or multidrug resistant transport protein. So if you are trying to treat a tumor with a drug, and then that tumor pumps the drug right back out, there's no drug left in the cell to kill the cell. So that cell that pumps out the drug becomes resistant. A similar thing can happen in bacteria. So you start treating a bacterial infection with an antibiotic, and that multidrug resistant transport protein pumps the antibiotic right back out. So that bacterial infection keeps growing. Something that's interesting here is that a lot of MDR transport proteins are able to transport a big variety of very dissimilar drugs, which is very different from most active transport that is very specific. So if we back up and remember that cystic fibrosis transporter from our facilitated diffusion discussion, the cystic fibrosis transporter, which allows ions to function, to flow through, keeps mucus from building up inside of your lungs. It has a lot of similarities to the core structure of that ABC transporter, but a big difference here is CFTR does not use ATP to drive transport. It's an ion channel that allows ions to flow through. It does use ATP to open the channel. Indirect active transport, or secondary active transport, is not going to be powered by ATP hydrolysis. It is going to be allowing one molecule to come down the concentration gradient while something else goes up. So here we have two molecules going through. These are symporters. One is going down their concentration gradient, and the energy that produces allows the other molecule to go against its concentration gradient. Or here, we're bringing sodium into the cell, going down its concentration gradient, to allow calcium out of the cell, going up its concentration gradient. So you're actively transporting a molecule against its gradient because it's coupled to something else going down their gradient. So none of these have an ATP binding area on them because none of them have to bind to ATP. None of them have to use up your ATP stores. We are just taking one thing down its gradient, and that gives enough energy to let something else go up. A lot of cells are continuously pumping sodium ions across our cell. If we're looking at animal cells, or protons across the cell if we're looking at other types of cells. So in animals, we have the sodium-potassium pump that keeps a concentration of sodium outside of the cell and potassium inside of the cell. So having that high level of extracellular sodium means you can bring some of that sodium back into the cell and allow something else to come in with it. So that movement of sodium down its gradient into the cell allows you to also bring in sugar or amino acids or things that wouldn't otherwise be able to come in. So one example would be the sodium. Actually, I'm going to come back and talk about the sodium-potassium pump in our next video. We have kind of hit our limit here. So I'll see you back to talk about that sodium-potassium pump in our next video. Bye.